Buenos días para todos. Les damos la bienvenida a esta charla programada en la mañana de hoy desde la Facultad de Ingeniería Ambiental. Es un placer pues tenerlos aquí a todos conectados, los que estén conectados. Esta charla pues no solo es diseñada para los estudiantes de Ingeniería Ambiental, de la Facultad de Ingeniería Ambiental de la Universidad Pontificia Bolivariana y especialmente a mis estudiantes del curso optativo de Sustainable Soil Management, sino que también está diseñada pues para todo público que quiera de pronto pues aprender un poco más sobre la importancia del suelo y la contaminación también que se da en estos espacios en el suelo precisamente. Y por eso en el día de hoy tenemos a una invitada muy especial. Su nombre es Carolina Novak. La doctora Carolina Novak nos va a contar un poco acerca de la contaminación en el suelo por plaguicidas y por qué es un desafío ambiental creciente, especialmente en nuestros ecosistemas agrícolas, en nuestros pues, países en estado en vía de desarrollo, en los que nosotros aún utilizamos gran cantidad de estas sustancias químicas. Exploraremos este papel fundamental que desempeñan también los microorganismos en el suelo y en la degradación de estos contaminantes que encontramos de una manera pues casi constante en el suelo. Y vamos a detallar especialmente la biodegradación de plaguicidas, por ejemplo, como el glifosato. Voy a cambiar un poco a inglés para hacer la presentación de la doctora Carolina Novak. Ella nos va a hacer su presentación en idioma inglés. Dr. Carolina Novak began her studies in environmental protection at the University of Warmia in Missouri in Poland, and she earned the degree in engineering in 2002 and the diploma in engineering in 2004. Then she received a scholarship in the Department of Agricultural Chemistry and Environmental Protection, and then she got a scholarship for a project like Aquabase in Germany. She obtains her doctoral degree from the University of Aachen in Germany in collaboration with the Helmholtz Center of Environmental Research in Leipzig. And then she got her habilitation at the University of Berlin. In recent years, she has been working as a researcher at the Helmholtz Center for Environmental Research in Leipzig, focusing in her work on the biodegradation of pollutants in relation with the non-structural residues and the impact of pesticides in soil. She has conducted innovative studies, the degradation of glyphosate, 2,4-D, and its influence on agricultural systems, and emphasizing the role of microorganisms and the formation of non-structural residues. Her research also is advanced in the prediction of the fate and biogenic residues, application and formation in soil. And today she is going to talk about the soil microbial activity and the pesticide biodegradation. So welcome, dear Dr. Carolina Novak, to our space today.
Okay, thank you very much for this introduction. So I will just start, and as this basic focus is on the microbial activity and pesticide biodegradation, as it was mentioned. And I will talk a little bit about biodegradability testing, how we can do this, and using the stable isotopes. So I guess probably all of you heard about so-called Sustainable Development Goals. So this is quite important now. So, and also pesticides also are part of this because they also end up in the environment. So, and one of this main overarching goal of this is of the Sustainable Development Goals is to guarantee good soil health, because it's needed. Good soil health is very needed to provide ecosystem service delivery. And that's, for example, there are different services, but for example, here I highlighted, for example, that we need to secure food and water. And the question is now, how is this security of food and water affected if the pesticides are ending up there? So, and this is the major part which I want to start. So, as we know, the pesticide pollution is a really problem globally. And if you look in the graph, for example, the total pesticides which were used in million tons. So, for example, from the 1990 compared to 2020, the global pesticides use doubled. So, and this is quite alarming. So here we can see the different these pesticides used in tonnage also depending on the continent. And you can easily see here marked with the red cycle, for example, that the South America is leader in the using of the pesticides in the world. And what else is also here, I want to highlight that this percentage use compared to 99 to 2020, it means roughly 20 years, increased more than 120%. So that means that the pesticide pollution is a really growing problem, and also especially in developing countries like South America. So also this is quite recent study, which was published by groups from Australia. They tried to make modeling group approaches to calculate the percentage risk on the global scale, and they ended up by calculation that 64% of world farmland is at risk of from the pesticide pollution. So, for example, if we look in the Latin America, which is actually calculated together with Caribbean coast, is, for example, with the circle, which is depends, the risk is higher when this orange intensity is stronger. So, for example, from the stronger to weaker, we see that the risk is quite high and medium risk is at least 60%. So that's why that means that we are really exposed to pesticide pollution as well, everywhere, basically in the whole world. So, and what that means, the risk from the pesticide pollution. So, for example, when the pesticides are sprayed on the crops, for example, to kill weeds, for example, what can actually happen with pesticides when they are after spraying? They can, for example, be taken up by plants, metabolized in plant tissues. They can be degraded by microorganisms. Also, they can be also sorbed to soil particles and get attached to that, but of course they also can be desorbed from soil and then leach to the groundwater. So, and what that means that, for example, at the very end, for example, the food which is contaminated with pesticides, or even fish, for example, which is swimming in the contaminated water, also can be end up in our body. And this is also quite a problem, and which can affect only our living organism. So how we can eliminate or at least minimize this risk pollution? So this is only what you see in the slides is on the European scale. So, and Europe decided to introduce some regulations to control this pesticide pollution as much as possible and to prevent the risk for the human and environment. So what that means, for example, the pesticides industry, which want to market specific pesticides, for example, glyphosate is named Roundup. So, and we have here companies, like, for example, I listed the most famous companies. Probably you heard about that. It's Bayer Crop Science, Corteva AgriScience is American, and BASF. So if they want to market pesticides first, before to get a permission to market these pesticides, they have to test this active compound and prove that this active compound is safe for us. So, and for these regulatory issues, there's authorities which are responsible for that. And for example, we have in Europe regional level, for example, like I'm now from Germany. So this this is the governmental institute, which is called Environmental Protection Agency. It's also responsible for the pesticides regulation in Germany. And on the Europe, it means European Union level, we have European Chemicals Agency, which is also establishing special guidelines, what should be done, how much tests should be done. And this agency, which is responsible for European Union, is based in Helsinki in Finland. So what that means with this regulatory testing, there is a specific guideline, which is very huge, over 700 pages, which is called Registration, Evaluation, Authorization, and Restriction of Chemicals. Is called, in short, REACH. And in this document, they always specify what actually has to be done, what should be tested, which properties. So, for example, I listed only just 3, the most important ones. For example, physical-chemical property of this active compound, whether it's toxic for human and aquatic life, and environmental fate, which one of this is biodegradability testing, about which I'm gonna to talk in more detail today. So how this biodegradability testing is done? So this is, you can see this slide with bottles. So for example, for that we use isotope. So isotope, with this isotope we labeled with heavy isotope because each element has, nearly each element has some kind of tiny amount of heavy isotope because they always occur in the mixture. So for example, I listed 2 isotopes, which I will show today. So for example, hydrogen, the most abundant hydrogen isotope is nearly 100%. And the hydrogen isotope has a very tiny percentage, 0.01% of the heavy isotope present. And this heavy isotope can be used to label our chemical, to which we add to our bottle with soy. So another isotope which is commonly used is a carbon. This is the most commonly used. It's nearly the most abundant isotope is 99%, and the heavy one, which we can use for labeling, is 1.07%. So, and using these isotopes, we look into specifically in the enrichment, and we quantify this isotope in the sample. And using these isotopes, we can, for example, look in the fate processes in soil. So as an example, I show glyphosate molecule. You see now, for example, the carbon labeling, which is marked with the red color. And this is the glyphosate molecule, which is labeled, for example, in this carbon position at position 3, or for example, position 2. Similar, this is hydrogen. It means heavy hydrogen, which means here this heavy hydrogen is deuterium in short. It also can be labeled to carbon, can be bound to carbon. So in the same position, here we have carbon, and here we have 2 hydrogen which are attached to carbon. So, and such a molecule which is labeled, we can add to soil and then just incubate and see what happened afterwards in the lab scale, because we can control conditions more under such a condition. So what actually we do during such a biodegradability testing, this is example of carbon isotope. So we see, for example, glyphosate, which is labeled with carbon position 3, we add to our soil and incubate for some time. And then during the incubation, we determine so-called carbon mass balance. What this carbon mass balance is. So we have, for example, mineralization. That means is how much of the compound was transformed to gas. It's carbon dioxide, which is trapped in sodium hydroxide trap. We can then, after incubation, take a soil and, for example, put some solvent, for example, aquatic, organic, and to try to take out as much as possible compound out of soil, which means can be extracted to the liquid. And this part of mineralization and extractable contaminant is quite easy to analyze because we separate from solid matrix and we can use different chromatographic devices. But the most challenging part of this biodegradability testing is the part which remains in the solid after extraction. So we remove solvent and then we have soil which still contains some labeled after extraction. And this part is quite challenging because this is labeled which was bound to soil. And this label is mostly quantified because it's difficult to characterize using standard methods. And those residues are called hidden residues. What these hidden residues can be? So we have 2 options. So for example, we see this red. This can be xenobiotic residues. That means it's pesticides which was not transformed, or its major transformation products, for example, can be bound to soil. and those residues can be potentially hazardous. The other option is green, you see, is a biomass residues. And those biomass residues is just basically compound which was degraded by microorganisms. They use this element to incorporate in biomass, and then after the microorganisms starve, they will be fixed in soil and they are harmless. So let's start with the xenobiotic residues, which are soil-bound xenobiotic residues. As I mentioned, they can be potentially hazardous. The risk depends how strongly they are bound to soil. So we see now here these 3 boxes. So, and the binding mode, for example, the compounds can be adsorbed, and depending, can be weakly adsorbed and can be strongly absorbed. And they can be released with extractions, which can be soft, can be strong, depends if we use just basically shaking, or for example, we use in addition to that, for example, pressure, temperature, very high temperature to take out. Another type of residues is physically entrapped. That means they are basically blocked inside the aggregates. And then for that, we have to disrupt the aggregates and using different methods, which I listed, for example, silation. And the last one is chemically bound. You see now, for example, the red color, this is the binding mode with pollutants and organic material of soil. So, and those ones usually are strongly bound. So we see the binding strength for the adsorb is quite weak to the highest when it's chemically bound. And of course, with that, depending how strongly they are bound, the compounds which are immobilized and so can be released. So for adsorb can be released quite quickly. And that's what that means for us. For example, we have sorbed chemical here, let's say in sediment, for example. And then we have, after certain conditions, can be released. And then after that, this compound can be taken up by fish, which we can consume. And then this can delay risk because the compound can later end up in our body or aquatic animals. So, and also this risk of the soil-bound xenobiotic residues can be even much higher, especially when we consider this climate change issue. Because according to this graph, we see the number of events globally. So there's flat, and which is the pink color, and flat with the blue color is increasing over time. And those conditions can even promote release of those bound chemicals. And this is also an issue which is quite alarming. Now we come, what is this soil-bound biomass residues, how they are formed? So for example, here we have a compound which is degraded by microorganisms. They use elements. So for example, here we have example. Can be pure plant material, which you see in the field, like leaves, for example, and pesticide. So, and any of them can be used by microorganisms to grow. For example, they take up carbon because we have to make our body. So microorganisms also can they need also some elements to make a body. So, and then they grow. After that, certain time when the substrate is gone, they start to starve. After then, they decay, and then simply the fragments are just suspended in soil. Here we have an example of the microbial biomass residues under scanning microscope, electron scanning microscope. And then here we can see, for example, with the red color and the red circles, you see this is the biomass residues after a certain time. And living cells are marked with the yellow color. So you see, so for example, here we have cells, living cells in batch culture. So it means the medium, we put microorganism medium, and here is the living organism, and this is the biomass residues in the medium. So here is the same in groundwater, in situ microcosm, we also have the same. We also see biomass residues here also in such a microcosm. And at the end, also we have soil microcosm. You see also where biomass residues also are released in soil. And all these fragments are of the cell envelopes, which basically become organic material at the end. And this organic material can be these biomass residues, which are harmless because this is just biomass residues. They are biological molecules, like, for example, amino acids, fatty acids, and so on. That's why they are harmless. So they either can be fixed biomass residues or recycled by other microorganisms, like a cycle. So, and how we can analyze this biomass residues in our soil sample? We can do this because we know basically how the microbial cell is built up. So for example, here is the microbial cell. So 70%, of course, the living biomass is mostly water, and 30% of the biomass cell is chemical. And what is the chemical part in this, which we can use these chemicals to analyze biomass residues in soil. So, and then we have different biomolecules we can choose which you want to analyze. For example, the major pool is proteins, and there are some other small molecules like ribonucleic acids and DNA, phospholipids, and so on. So here I have a table. For example, here we have bacterium and yeast. It's basically the values are quite comparable. But for example, here we have, of course, as I mentioned, water is dominant in living biomass, but after the biomass die, the water is gone. And then we have the dry weight, which is the most important part, which we can quantify biomass residues. For example, we decide to choose proteins, and the monomer of proteins are amino acids. So we can just extract amino acids because they are the major component of biomass cell, especially for the chemical composition. So it's around half of the cell carbon, for example, is around 50%. We can also analyze other components, nucleotides, for example, which also can have a contribution up to 23% from DNA, RNA, also fatty acids. Today, I will show application of amino acids and fatty acids. So let's start with the fatty acids, which is called phospholipids fatty acids. So we have what we can do. We can get such information, for example, about the 4 microbial groups when we analyze these fatty acids in soil. For example, we can analyze gram-negative bacteria, starving gram-negative bacteria, gram-positive bacteria, actinobacteria, which is gram-positive phylum, and fungi. So here we see, for example, the fatty acids. We have, for example, if we have like branch fatty acids, this is typical for gram-positive bacteria. Like, for example, here is example of actinobacteria. If we have 1 double bond, then this indicates gram-negative bacteria. If we have 2 double bonds, then this is fungi. And we have the triangle in the fatty acids molecule. That means this is starving gram-negative bacteria, which is coming from here. And then they start to increase rigid to make it more resistant to degradation because they are starving. So based on the fatty acids, we can check, for example, microbial activity. And for example, what are the microbial degraders based on this phospholipids fatty acids classes? And then we can simply identify 4 groups of degraders. As an example, okay, and this is the more or less the workflow. Usually we analyze fatty acids combined with isotope labeling. So as I mentioned, we use isotopes to distinguish the degradation process, because we also have also phospholipids in the soil which are unlabeled. And that's why we need to label our compound to be able to see really phospholipids fatty acid which comes from our compound, not which is occurring in soil naturally. So for that one, we always make an incubation experiment in which we add unlabeled compound, for example, glyphosate, and then labeled glyphosate, because we need to always compare 2. After that, we destruct cell membrane, because we have to take from living biomass fatty acids out. And then we have fatty acid, which is not labeled, with a control sample, and labeled sample, which has our control fatty acids plus labeled fatty acids. And then we can also purify fatty acids and derivatives and analyze using gas chromatography mass spectrometry devices, for example, to quantify with gas chromatography mass spectrometry, and gas chromatography isotope ratio mass spectrometry, which is also helping us to distinguish between 2 different isotopes, to separate isotope in our sample. So as an example, this disappeared completely. It is conversion. I cannot show that actually graph, but what was the option behind of this? Because it's during the conversion disappeared the graph. I didn't expect that. in this case, we usually can see that in this graph, we could see that fatty acids, all different label groups, all different groups like fungi, gram-negative bacteria, and positive were labeled, because usually the situation is like that microorganisms start to degrade, they are starting degraders, and then they die, and then the other groups or microbial groups take over label from initial degraders. So, for example, we have the metamitron. This is a pesticide which was labeled. Microorganisms started to use this, to grow on that. They decay, and then the part of this residues was stabilized in soil, and part can be recycled by other microorganisms, and then as a cycle. So that's why the fatty acids usually are labeled between different microbial groups because of passing label between different groups. So another biomarker, which I mentioned, which is quite dominant biomarker in the microbial cell, is the amino acids. We have different type of amino acids, which depends on the, for example, polarity of the, for example, we have neutral for the amino acids, acidic amino acids like glutamate and acetate acid, and also we have basic amino acids. So based on the extraction of amino acids, we can quantify roughly how much biomass residues were formed from pesticides biodegradation. So because we know that amino acids can constitute to up to 50%, 55% of total chemical pool of the microbial cell. And if we multiply amino acids content by 2, we can calculate more or less how much biomass residues were formed. So, and here we have the amino acid stable isotope probing is similar way like fatty acids. The only difference is that the procedure is a bit more time consuming because we need to break bonds, peptide bonds in the hydrolysis process to release amino acids from proteins. But the overall process, like purification, derivatization, is quite similar procedure and analytics as well. So here we have example of the application of labeling. For example, you can see the glyphosate, which was labeled with 2 position carbon and 3 position here. So, and then we can try to calculate the percentage of the initially labeled in our system, which I showed at the beginning. So, for example, how much was of the labeled was released as a carbon dioxide. So in both cases, we can see, for example, it's 30% for labeling position 2, and for example, 40% when we have position labeling number 3. So we also have, after that, we also try to extract soy, from soy the chemical, it means glyphosate and AMPA, for example. So in that case was around 7%. In that, for the labeling position 3, we have extracted chemical was a bit higher, was 28%, because the transformation product, which is called AMPA, is in that labeling. When we label this molecule, it will be AMPA will be labeled compared to glyphosate 2, which AMPA will be unlabeled. But and the amount of amino acids which could be used to quantify biomass residues is basically only 11%, which is green for labeling position 2, and only 2.2% for labeling position 3. But we see this gray boxes in both labeling in these 2 graphs is pretty high, is 48% unknown residues and 28 also another unknown residue. Still, we don't know what is that. It's quite challenging to identify that. So to summarize this part, I just want to highlight the application of biomolecules in biodegradability testing with isotopes, for example, when we analyze, for example, phospholipids, fatty acids from soy, we can distinguish between different structure of the fatty acids, between the different groups of microbial degraders, 4 major groups. We also can distinguish between living and dead biomass, and also amino acids, which we can quantify the total amount of biomass residues, because if we by extracting amino acids, we can just multiply this amount of amino acids which we extracted by 2, and then we get the total amount of biomass residues which were formed from degradation from degraded pesticide. So, as I already mentioned, this type of residues which are remaining in soy after an extraction are quite challenging because they are quite hidden and not easily accessible for analytics. So, for example, as I mentioned, these xenobiotic residues, we could try to cleave or extract some sorb chemicals, but the analytics can be mild, harsh, or even destructive. And also we have the biomass residues, which also can be analyzed, like I mentioned, for example, amino acids, but the laboratory work is really time-consuming and very expensive. And this analytics is not really satisfactory option for industry company because they have to test a lot of chemicals within a short time, and they don't have much time to test each separate compound in very detail. So the concept is now how we can simplify the distinction between these two types of residues more efficiently, so by saving time. So therefore, I just also suggested with a new idea, new concept, how to distinguish between these two different types of residues. I applied for this Innovative Science Award. This is the Chemical Industrial Council, which is based in Brussels, in Belgium. And I applied for that and suggested a new completely concept, how we can distinguish these residues more efficiently. So I suggested to use hydrogen isotope instead, for example, of carbon. Because when we use hydrogen isotope, the biomass residues will be minimally formed and mostly will be xenobiotic residues. So why actually is happening like this? Because we just look in the labeling cycle of carbon and hydrogen, which is nearly comparable. So as I mentioned, for example, we have carbon labeled compounds, which means carbon from that molecule can be used by microorganisms. They can be simply passed to another this label can be passed to another microbes after this primary degraders die, and then the label is recycled and only slowly released as a carbon dioxide. So therefore, we have quite high amount of biomass residues when we use carbon labeling. If we compare to hydrogen, basically the cycle is exactly the same like carbon, but with hydrogen, the situation is already different at the very beginning. For example, when this compound is degraded by microorganisms, then this hydrogen, which is attached to carbon, get released to water first, to soy water, and then it gets diluted with soy water, and then only hydrogen from water is taken up by microorganisms. So that's why we have huge dilution at the very beginning, because of this dilution at the very beginning of degradation, if the compound is degraded. So, and it was the concept behind that. So, and to check that, we made also some experiments. I show only just 2 examples. For example, here we have labeling of the 2 compounds. For example, with the red color is the carbon compound, is a 2,4-D-chlorophenoxyacetic acid. It is a common herbicide. And here we have labeling carbon in the aromatic ring. But with hydrogen, we have the hydrogen attached to carbons in the aromatic ring to mimic the same labeling position. So, for example, here we make the same for glyphosate labeling position. Here is the label carbon at position 3, is a 1 carbon, and to that carbon there are 2 hydrogen attached, so with heavy hydrogen. So, and we just added such a molecule to soil and incubated and check how much it will stay in the soil after extraction. So, and then we quantify total amount of label in soil after extraction. So we analyze how much amino acids got the isotope label to quantify the biomass residues. We also quantify total amount of residues which could be unknown residues. That means total amount of label present in soil minus biomass residues. And this can be basically two types of residues. And the idea behind is that overarching question is, can we skip hydrogen labeling and analytics of the biomass residues for hydrogen labeling? Because we expect very minimal amount of biomass residues form when the hydrogen is labeled substrate is added to soil. So here we have the comparison of two isotope labeling. So for example, we see this is the percentage of applied isotope label. And here we have 2,4-D molecule. And here we have the biomass residues which comes from 2,4-D label with carbon and with hydrogen. So if we look at these two green bars, we can instantly see that, for example, for carbon, we have around 20% of the bound residues. But if we compare this total amount of bound residues for hydrogen, it's below 5%, it's around 4% only. So this is the difference factor of 6%, 6 times less. So for example, situation for glyphosate position 3, here we have carbon label. For example, here we have similar situation as for 2,4-D, that around 32% total amount of residues were formed compared to hydrogen, which was 15%. So, and the biomass residues, for example, also was quite low for carbon. It was around 4%, but for hydrogen was even lower, so it's 5 times lower. So based on this study, we could definitely prove that the amount of biomass, the amount of hydrogen, which is incorporated into microbial biomass, is very low if we compare to carbon because of this dilution, which I mentioned with water. So therefore, if we use, for example, hydrogen labeling, mostly the residues will be xenobiotic residues and maybe very tiny amount of biomass residues. And in that approach, we can just quantify the total hydrogen label without going into detail and skip analytics of the biomass residues. So we also proved that for other compounds, but I show only just 2 today, just for sake of simplicity. So based on that, we can draw conclusion that hydrogen isotope in biodegradability testing part, as I mentioned, if we use carbon isotope labeling, you have 2 type of residues which need to be characterized, which need to be identified. It means potentially hazardous xenobiotic residues and harmless biomass residues. In case of hydrogen, the biomass residues will be minimally formed because of this huge dilution, and most of the residues will be xenobiotic residues, which we can just quantify. And this makes much easier for companies, especially when they have to test many compounds. So therefore, for future biodegradability testing, like for example, for company like Bayer Crop Science, which I already showed them this idea for Bayer company, but also other companies that they could use hydrogen to exclude biomass residues formation. And in case if the compound is not degraded, for example, the hydrogen will be simply bound as xenobiotic residues, and we can simply quantify them. And then with this, we know directly whether we have xenobiotic residues or biomass residues when we use hydrogen isotope. So this is quite new approach, and we have already some proofs, but still want to prove for radioactive hydrogen, so which is still pending. So, and this is all what I presented today. So I want to thank you for your attention. So you are free to have questions to me. I also have some stimulating questions if you want to hear from my side, if you want to have some stimulation for discussion. It's probably maybe not very difficult, but it's very general. So is, for example, one could be, is the pesticide application controlled, regulated in Colombia? And what you could do to control pesticide application and pollution in Colombia? So thank you very much. So, and I invite you for discussion.
Thank you, Carolina, for a very interesting presentation. Here in Colombia, actually, we don't have any control or regulated application of pesticides, which is something that we have to address in the common future. I know that in some countries you have this kind of regulation, especially in the European Union, where they take some samples in soil and they control in all parts of the of this regulated crops they are taking from the farmers, right? So this is really common in the European Union.
Yeah, I mean, we also have in the US also has some regulation, which is a little bit different, but it's quite similar to our regulation in Europe. But that means that, for example, if you have like a food imported, for example, from Colombia to other Europe, so we don't know, for example, it might be potentially contaminated too. So is there any
I
option
think for regulation of for export, exportation of sport, exportation of food, the regulations are different because then they have to check the regulation of the country that where the food is is traveling to. So I guess if we want to cultivate some goods for European Union, we have to fulfill all the requirements for this exportation. So I guess this will be controlled. But inside our country, for internal distribution, I don't think that is well regulated.
Mm-hmm. Yeah, this could be. Yeah, but then maybe this is also good room for improvements to convince the authority to maybe set up such a regulation, maybe to test a bit for people to protect people in Colombia, maybe inside the Colombia, for
Yeah,
example.
because now I guess the people is more aware of the problems that many pesticides can cause in their bodies, in their health. So now they are checking and they are consuming more products that are organic and they proceed from organic source. So they are more aware of all this information because the globalization and all the news that we have already in the net, and the people is informed more about all these troubles and problems. And so I guess they have more conscience now than before. So, but still we have a large room to explore and to get in order to have a more efficient system that we can control this pesticides application.
Yeah, I think this probably will need some time, I guess, because usually for REACH regulation, it was, I think, nearly 10 years to create this regulation in Europe. It was nearly 10 years.
Yeah, we
And
have
it
to
was
start.
launched 2007. This
Yeah, for
takes.
example, in Germany, you have this regulation that protect the soil. This regulation, um, like only for soil, and here is something that we miss in Colombia that we don't have. I guess this will be a good idea to address in the future. So I get some questions from the audience. They are asking about the implications of the research that you already presented on the regulatory practices and the environmental monitoring, because you were talking about industry, but how about these regulations in the European Union or even in Germany, how they can apply this kind of technology or new research findings?
Yeah, I mean, usually basically for carbon, isotope is kind of standard. And there's also new regulation. They are really applying that. They really test this, what I presented with carbon, like mass balance, they do this. And for also they have to do more tests for this to distinguish between residues, type of residues. They have to do it. There's a like huge document, which was actually written by Matthias Kissner, Stefan Trapp, and Andreas Schäfer, actually, and they have this huge document to specify how to do it. For carbon, yes, we have to do this, and this is really used in practice, really used. For hydrogen, this new approach, not yet. It will take time, because for this biomass residues idea, which started in my PhD time, it also they were not considered in regulatory testing. So it was basically, the idea was published in 2011. So, and it took them 7 years to convince them and to implement this analytics of biomass residues on the European level. So this is already used. For hydrogen, it will take time because it's extremely new approach, which just was published, just accepted in Nature Communications now. So of course, I mean, it's still too new to convince. That's why I need to test this with radioactive hydrogen, because they use labeled compounds for that, because radioactivity is more commonly used for regulatory purpose, not stable isotopes, because stable isotopes is really time consuming. And yeah, not everyone's experienced with this machines, operation, and so on.
Yeah, and how expensive it will be to implement this kind of testing. I mean,
For hydrogen,
yes.
yeah, I mean, that's a good question. But I mean, the problem is with hydrogen, because with heavy hydrogen, stable hydrogen, you can get compounds easily on the market because they are used in the chemistry as a surrogates, as internal standards in chemistry. But tritium label, which is equivalent to radioactive hydrogen, is not. And I think the biggest problem is to get this labeled with tritium. It's not difficult to label that, but on the market, you cannot get that. But I believe that if you convince company that tritium works, because I want to test with company now, with some compounds, with tritium, they can label by themselves, because usually the company label these molecules by themselves. They have like a chemistry division in their company, and they label molecules by themselves. So, and they label carbon by themselves. They don't buy from chemical company. They just make compounds by themselves. And hydrogen is much easier to label than carbon. It's much easier. Of course, you need some experience because you need some metal catalyzers, but you can do it. But first, I have to convince them that it makes sense to do that. So, I mean, for hydrogen, stable hydrogen is convincing. And I already I'm in discussion with Biocrop Science and some other people to test with tritium, if they can test this, because I cannot do this. So if we can prove that, maybe we can implement this test, but it will take time.
It will take, yeah. I have a question of the audience, and it says, "Hi, I would like to know from Valentina Jimenez. I would like to know what are the most important regulations for crops in Germany."
I mean, for crops, I mean, in terms of pollution,
Yeah,
or
for pesticides, pollution.
yeah, I mean, basically for crops, I don't think they investigate really on crops. Like, you know, maybe they have some specific, like a threshold limit, you know, like for example, you take your food and you just simply extract chemicals with solvents and analyze, and you have like an upper limit, which is permitted to be launched on market. They make some tests of this, but of course, I believe it's difficult to test all food like this. Yeah, so I guess they make it randomly somehow. Yeah, so and we also have this biofood, which is called biofood, organic food. And for the organic food, they test more than non-organic one. So usually they have some threshold level. Yeah, so which is also another authority responsible in Italy for that, and they test
Yeah,
this.
yeah. It's depending on which kind of pesticides you are using.
Yes, but I think this, I don't
It's not the
remember the
same for
threshold
each
number.
pesticide.
Yeah, but usually it's comparable to pesticides. In drinking water resources, we have threshold for drinking water and for crops. I don't remember the values, but this basically for pesticides is more or less the same number as far as I remember, at least for water.
Okay, there is another question about how the findings of the research can be applied to mitigate the risk in agricultural systems. Because you talk about companies, we talk about maybe regulation, but how we can send this information to mitigate this risk of pesticide contamination in agricultural systems?
Yes,
How we
I
can
mean,
avoid this?
yeah, I mean, the problem is that usually we test in the lab because we can control that. Yeah. In the agriculture field, it's more difficult because it's huge. You would have to take thousands of samples. Yeah. So you cannot really 100%. So the idea behind this regulation is to simply test, prevent having problem in the field. So that means we test in the lab and prove, okay, is biodegraded pesticides, then we can launch to the market because in agriculture will be okay. We consider worst case scenario in real lab, but of course, agriculture might be slightly different. Because now, I don't know if you heard about, there is also now discussion to change incubation temperature. Not 20 degrees, but 4 degrees now for regulation. Because in Europe, you don't have 20 degrees all year like in Colombia. We have 4 degrees as an average. Yeah. And lower temperature, of course, it means slower degradation. So.
Okay. And finally, another question is how emerging contaminants or new generation pesticides differ from traditional ones in terms of biodegradation. What do you think?
The emerging one? Yeah, I
Or the
mean,
new pesticides, there is a difference between the pesticides that are maybe they are processing and launching for the market today in comparison to those that they were maybe using in the past. What do you think?
usually it doesn't matter if it's emerging, for example, because what they do basically in the regulatory perspective, they get permission to launch active compound. Doesn't matter if this is new or emerging one. So they get like a 5 years time. And then after this license like expire after 5 years, you have to launch again, apply again for old one and for new one. So basically they are treated equally. Yeah, of course, emerging one, you need to do more tests because we don't have any more data available in publications. Yeah, because with old one, they can cite publication or we got this data in our lab, and there are also other proofs from other studies published in that journal and that. Emerging, not. That means they have to do more tests. But anyway, if you get permission to launch your pesticides, it's not forever, it's just 5 years. And then after 5 years, you have to again make testing. And usually within 5 years, we have new tests. It's not like always the same test. Every 5 years, they always ask for more. Not less, but more. And so that's why always they have to renew this permission.
And do you think these regulations are getting more strict or not?
It's much more strict now. They want to do human tests in the human cells, like produce artificially in the lab because they don't want to test on the animals, because they want to save like a mouse animals. They don't want to stress animals. They want to have cells now, like artificially produced in the lab. They want to save animals. They ask for more tests, more properties. Temperature, this is the new regulation, which will be 4 degrees, as I said, that will be new. So usually it's more strict, it's much more strict, and it's more to do, always more to do.
Okay, thanks a lot, Carolina, for this interesting talk of today. I hope that we can invite you in another opportunity for more information about pesticide in soil and microbial biodegradation of pesticides in soil. You are really welcome to our platform and also to our university every time. Thank you very much for this interesting talk.
Thank you very much too.
So with this, we close our talk of today. Damos entonces cierre a nuestra charla del día de hoy. Damos las gracias a la presentadora Carolina Novak por esa interesante información y a ustedes pues por vernos y hasta una próxima oportunidad. Hasta luego.